Good morning, everyone. My name's Professor Sarah Copeland. I'm the President-elect of the Pathological Society, and I'm standing in for Mark Ahrens, who is in Tasmania. I hope the weather's been good for everybody so far, and I'm welcoming you all to May's PathSoc Grand Round Lecture. These Grand Round lectures are sponsored by the Shanks Foundation, which were established by the late Shanks, who was a pathologist based in London. And our speaker this morning is Professor Gisli Jenkins, who is an NIHR research professor and holds the Margaret Turner-Warwick Chair of Thoracic Medicine at the Imperial College, London, and is also associated with the Royal Brompton and Harefield Foundation Trust. He's the head of the Margaret Turner-Warwick Centre for Fibrosing Lung Diseases and has a very strong track record, research record in this area of interstitial lung diseases and pulmonary fibrosis. He's been involved in a number of longitudinal observational studies and is the lead in the GSIP partnership in respiratory medicine following the Genomics England 100,000 genomes, and has been involved in a number of studies related also to COVID. He's received a number of awards, which include the European Society Gold Medal in Interstitial Lung Disease in 2020 and the British Thoracic Society's Meritorious Service Award in 2022. So it gives me great pleasure to introduce Professor Jenkins, who will be speaking today about interstitial lung disease. Thank you very much.
Thank you. Thank you for that kind introduction. And now I'm going to see if I can share my screen.
I should just say to everybody that there is a Q&A button at the bottom of the screen. Please put in your questions and we'll answer them at the end of Gisli's lecture. Thank you.
So hopefully you can all see my slides.
That looks good, Gisli, thank you.
Okay. So I'm going to talk about the molecular pathology of idiopathic pulmonary fibrosis. So I'm going to talk briefly about idiopathic pulmonary fibrosis because I'm assuming that many people don't have a detailed understanding of this. And then I'm going to talk about alveolar homeostasis and injury because these are crucial precursors to the development of pulmonary fibrosis. And then I'll describe some of our work, which has highlighted how this goes wrong and how this prevents normal alveolar repair and fibrosis, with particular focus on regulation of alpha-V beta-6 integrin-mediated TGF-beta activation, basement membrane integrity, and then show some of our more recent work trying to identify the fibrotic niche. So if we think about idiopathic pulmonary fibrosis, it's a chronic progressive disease of elderly people, often men. It usually presents with a 2 to 3 year history of progressive breathlessness and exertion and a dry, non-productive cough. And these symptoms are often very sort of insubstantial to begin with, and they're often misdiagnosed for heart failure, asthma, or COPD. There's often a history of smoking, normally ex-smoking, often 20 to 30 years prior to the onset of symptoms, and work in building and manual trades. Family history is becoming much more relevant, and I'll show you some genetic data. And it's a disease associated with aging and, as I said, male sex. And it's also associated with other fibrotic diseases such as systemic hypertension, type 2 diabetes, and ischemic heart disease. It has a dreadful prognosis. These are data from the Profile study, which is one of our longitudinal observational studies. And the median survival in our cohort was 1,284 days, which is just over 3 years. And not only does it have high mortality and poor median survival, but the journey from diagnosis to end of life is not a particularly comfortable one either. And as you can see from this picture in a support group, many of our patients will require oxygen for the course of their illness. It may be ambulatory. If you look at the back, these patients can walk, pardon me, but they require oxygen. And then as the disease progresses, they can do less and less for themselves. And as you see here at the front of the picture, patients are often in wheelchairs requiring 100% oxygen. And if we compare pulmonary fibrosis with various types of cancer, you can see that the 5-year survival is worse than pretty much all cancers, with the exception of lung and pancreatic cancer. And actually, it's about the same as with a 25% 5-year survival. It's about the same as esophageal and gastric cancer. And the incidence of pulmonary fibrosis is rising, and these are data published over 10 years ago now, showing a 6-fold rise in the mortality of pulmonary fibrosis from the 60s to the late noughties. And this has shown signs of acceleration over the last 10 years or so. So it doesn't look as though the epidemic is slowing down. And indeed, as we learn more about alveolar injury, I think the things like COVID and pollution are going to make the incidence of pulmonary fibrosis even higher. And as I mentioned earlier, it's a progressive disease, and this is illustrated beautifully in these CT scans taken from the native lungs of patients who'd had single lung transplants. And as you can see over the course of 4 or 5 years here, that the fibrosis, which is seen on this radiology, on the CT scan as these honeycombing areas peripherally, these progress remorselessly over time centrally. And if these patients hadn't have had single lung transplants, they would have certainly died before these scans were taken. So how does this happen? So you'll all recognize this. I'm sure this is a micrograph of normal alveolar structure, and it's stained with a Masson's trichrome. And you can see the alveolus here and the capillary here stuck together with the interstitial matrix. And this is represented by this cartoon. So we have the type 1 and type 2 epithelial cells, the alveolar macrophage, the interstitial fibroblast, and the dendritic cell. And where the type 1 cells and the capillary endothelial cells communicate, the basement membrane is shared. It's a delicate structure. 2 microns from the lumen of the alveolus to the lumen of the capillary, and it's easily damaged by things like oxygen-free radicals, inhaled dust. This was a pre-COVID virus and immune-mediated injury. And in the context of an appropriate genetic host, this injury can go wrong. And there are a number of different genes that we know that cause, that promote fibrosis. There are rare variants which are causal because the effect size is high, so the amount of injury required is less. And we describe rare variants as having a less than 1% minor allele frequency. They're normally in coding regions and are often associated with loss of function of the genes. In contrast, common variants are regarded more as risk factors rather than causal. So these are variants that require the presence of an environmental trigger. And the most notable here is MUC5B, which has an effect size of 5-fold for a single copy of the allele. But the vast majority of these common variants have an effect size somewhere between 0.5 or a doubling increase in risk. And these are generally in non-coding regulatory regions, but will have a minor allele frequency of 5% or more. And some of these alleles have a effect allele ratio of about 40%. So they're very, very common. And these are the alleles which will lead to idiopathic pulmonary fibrosis, as opposed to the rare variants which will lead to familial pulmonary fibrosis. And so when you have the appropriate environmental insult in an at-risk host, you get this scenario, which again, as pathologists, I'm sure you'll all recognize, this is fibrotic lung, again stained with Masson's trichrome, and the key features are illustrated here. So you get what we used to call bronchialization of the alveolus, but now we know these are transitional cells which are transitioning from AT2 to AT1 cells. And we'll talk more about those later. There is a fibronectin-rich matrix, infiltration of the interstitium with fibroblasts, which are either proliferating from fibrotic stem cells or have come from transition from alveolar cells or even from fibrocytes. There is some evidence of inflammation, neutrophils, activated macrophages, and lymphocytes within the lumen, and also apoptotic epithelial cells. And importantly, the basement membrane is disrupted, and we've long since known about this. The first descriptions of the disrupted basement membrane in IPF were in the 70s. So how does this happen? Well, one of the key molecules promoting fibrosis is transforming growth factor beta. It does all the things you would expect a profibrotic molecule to do. It prevents alveolar repair through a variety of mechanisms, and it promotes matrix deposition. And it's actually this little 25 kilodalton homodimer here, which is the active molecule, and it's kept in an inactive state through association with the latency-associated peptide and the latent TGF-beta binding protein. Now, there's enough of this TGF-beta preformed and sequestered in the extracellular matrix to turn us all to statues if it was active spontaneously. So this is why it has this very complex activation mechanism. And within the latency-associated peptide, there's an RGD motif. And this is important because it allows the latency-associated peptide to bind integrins. And integrins are cell surface heterodimers. There are 18 alpha and 8 beta subunits. 24 different combinations have been described, and 8 of these contain an RGD binding domain, of which 5 can activate TGF-beta. And I'm going to focus on alpha V beta 6 and alpha V beta 1. And so years ago, when I was a postdoc in San Francisco, I was sort of tasked with trying to understand how the epithelial alpha V beta 6 integrin was activated following injury. And we described a mechanism through which the cytoskeleton tugged on the cytoplasmic domain of the epithelial integrin, leading to a conformational change in the latent TGF-beta complex, which allowed the TGF-beta to interact with the TGF-beta receptor on the neighboring cells. Now, the important thing is that it requires direct cell-cell contact. And although the crystal structure has been solved and there is release of the active TGF-beta from the latent complex, its diffusion distance is tiny, so it has to be right on the receptor, and there are mechanisms which regulate that, and I'll show you some slides to that effect later. And so myself and my group members over the years have mapped the pathway which, by injury from things like lysophosphatidic acid thrombin, sphingosine-1 phosphate, act via their respective G protein coupled receptors to signal through a pathway involving G-alpha Q11, Rho-A, and Rho kinase to lead to myosin light chain activation and reorganization of the cytoskeletal structure, which then activates the integrin. And under normal circumstances, this then leads to SMAD phosphorylation in neighboring cells, and this is regulated by the ALK1 transcriptional repressor to keep the system damped down and activate just enough of the integrin to suppress alveolar macrophages. The TGF-beta acts on alveolar macrophages to keep them quiet. So TGF-beta is an anti-inflammatory molecule. It's a pro-fibrotic molecule, but it's an anti-inflammatory molecule. So the sort of homeostatic regulation is that you will, if the alveolar is moderately injured, it sets off this signaling cascade, keeps the macrophages under control. And importantly, you'll note that the basement membrane remains intact. However, we know that in patients with pulmonary fibrosis, there are very high levels of alpha V beta-6 integrin, and the higher the level, the worse the prognosis. And these were data that we generated a few years ago now from lung tissue sections from collaborators in Edinburgh, and the increased integrin on the alveolar cells was associated with a dramatically reduced prognosis post-biopsy. And there's a positive feedback loop. So if you stimulate an epithelial cell with TGF-beta, it will induce the expression of more beta-6 on the surface of the epithelial cell. And we showed this in human cells, but it's been shown in every species that has been tested. And within the beta-6 promoter, there's a range of repressors, and a number of them are shown here, and some interesting ones include the glucocorticoid receptor and the progesterone receptor. But the ones that I'd like to focus on are the ALK repressor. And when we transfected epithelial cells with ALK1 siRNA, we were able to reduce the repression. So when we treated these cells with TGF-beta, there was increased expression from the beta-6 promoter, which was driving luciferase. And similarly, when we measured the beta-6 on the cell surface of these cells, there was a slight but significant shift in the amount of beta-6 expressed on the surface of these cells following transfection with ALK1 siRNA. So if you block ALK1, there is increased beta-6 expression in epithelial cells. Similarly, if you knock out ALK1 in mice, they have increased beta-6 mRNA at baseline and in response to bleomycin-induced lung injury. Similarly, in these mice, when you give them a bleomycin-induced lung injury, which is one of our favorite forms of inducing fibrosis in animal models, they have an exaggerated response to bleomycin, suggesting again that not only does ALK1 repress beta-6 expression, but it helps to prevent the development of collagen deposition. And there is evidence that this pathway is involved in human disease also. Here is immunohistochemistry for normal lung, showing that there is very little beta-6 that is visible by immunohistochemistry. But in IPF, you see that the epithelium overlying the fibrotic regions here is highly expressing beta-6, which has been shown by many people. In contrast, ALK1 shows high levels of expression in normal epithelium, but in the epithelium associated with the fibrotic regions, it's disappeared. Similarly, ALK1 knockout mice also have evidence of spontaneous lung fibrosis if you allow them to age. As I mentioned earlier, pulmonary fibrosis is a disease of aging. And so we just generated these knockout mice and allowed them to live in their cages for a year. And at the end of a year, they were euthanized. And as you can see, they had these little patches of fibrosis, which were staining yellow and by stringent assessment of serious red. And there's a small but significant increase in global hydroxyproline content. And when we measured the mean alveolar wall thickness, there was a significant increase in the thickness of the alveolar walls in ALK1 knockout mice. So these are, and you can see here on the birefringent staining that effect. So it does appear that if you don't have ALK1, this transcriptional repressor, then you are predisposed to developing pulmonary fibrosis. And as I mentioned earlier, there are a number of genes that have been identified that promote fibrosis. And I'm going to talk about the first one that we sort of identified, which is ACAP13. So the genetic analysis highlighted that this was the sentinel variant here, this blue one. And when we did EQTL analysis, the sentinel variant was actually in between the ACAP13 gene and another gene, but it was associated with higher levels of ACAP13 mRNA by EQTL. And when we looked at the allele, the minor allele versus the common allele, we saw that with each copy of the minor allele, there was increased expression of ACAP13 in lung tissue. And this was in 3 separate cohorts from Canada and Europe. When we looked at the histology, what you can see is that in normal lung, ACAP13 is expressed in epithelium, particularly bronchial epithelium, type 1 and type 2 cells. But in patients with pulmonary fibrosis, there is high levels of ACAP13 in the epithelium overlying the fibrotic regions, not so much within the fibroblastic foci, but there is also high levels within lymphoid follicles, although we don't know what it is doing in those cells at the moment. And ACAP13 is a fascinating molecule because it's a scaffold protein that's crucial both for RhoA and pKa signaling. So LPA signals through GQ11 to RhoA, and ACAP13 acts as a RhoGEF, amplifying this signal. But it also recruits protein kinase A to counteract RhoA. So it has a sort of a dual effect, this balance, which maintains homeostasis under physiological conditions. However, if you have increased ACAP13, you will get increased potentially RhoA activation, which can lead to increased TGF-beta activation, and I'll show some data to support that. But it also has a PDE4 domain, which will inhibit cyclic AMP and protein kinase A, further activating Rho. And it's of note that there's a PDE4 inhibitor which has been developed by Boehringer Ingelheim, which is showing promise in clinical trials. So to understand the effect of the ACAP13 variant that we found in our genetic studies, we adopted an inhibitor approach, a siRNA approach, and a CRISPR-Cas9 approach to try and manipulate it. So using a fairly dirty inhibitor of ACAP13, we were able to reduce LPA-induced RhoA activation using a G-lyser with increasing concentrations of this ACAP13 inhibitor. It also led to downstream inhibition of phosphorylation of SMAD2, which is a readout for TGF-beta activation. And this is quantified here. We also developed a specific CRISPR-Cas9 strategy, one which targeted the whole of exon 5, which would wipe out all ACAP13 activity, and another single nucleotide substitution to put in the RS6202 variant, which we have associated with pulmonary fibrosis, and to see what effect that would have. And interestingly, so when we produce the exon 5 mutant, we do knock out ACAP13, and you can see here in this immunocytochemistry, there's very few cells expressing any green ACAP13. In contrast, when we mutate the RS6202 from wild type to the variant associated with IPF, you see a large increase in the number of shorter isoforms of ACAP13 and a much higher level of ACAP13 in the cells that are CRISPRed. And if I remind you of our schema of how ACAP13 interacts with PDE4 and cyclic AMP, you'll see that if we knock out ACAP13, you get increased evidence of cyclic AMP activity, which would be a good thing in pulmonary fibrosis. Whereas if we put in the mutant ACAP13 associated with pulmonary fibrosis genetically, you see there's a reduction of cyclic AMP, which would be a bad thing in pulmonary fibrosis. So if you mutate ACAT13, you can get profibrotic effects. So switching track a little bit, I'm going to talk now about the alpha V beta 1 integrin, because the alpha V beta 6 integrin is only found in epithelium, but we know that fibroblasts are important effector cells for promoting fibrosis. But the beta 6 integrin isn't found in fibroblasts. So how does this activate? How does TGF-beta get activated in these cells? And one way is through beta 1 integrin-mediated TGF-beta activation. So LPA, which we know signals through GQ11 and RhoA in epithelial cells, is also able to do this in fibroblasts. And you can block this with a TGF-beta inhibitor or increasing concentrations of the integrin beta 1 inhibitor, as shown in this Western blot. And so in collaboration with Galecto, we looked at galectin-3 inhibitors to see if they would inhibit this pathway, and indeed they did. And TD139 was one of their promising preclinical candidates, which unfortunately hasn't progressed beyond phase 2. But what we showed was that if you used TD139 to inhibit galectin-3, you were able to inhibit TGF-beta activation in fibroblasts pretty much to the same extent as an ALK5 inhibitor. And using more specific inhibitors, we got the same effect. Now, galectin-3 and 1 are ubiquitous molecules that are expressed all over the place, inside the cells, outside the cells, in the extracellular space. So to try and understand how the galectin-3 inhibitors were working, we used a galectin-3 extracellular inhibitor specifically. And again, we found that increasing concentrations of this extracellular galectin-3 inhibitor reduced TGF-beta activation, suggesting to us that it was an effect extracellularly. And so we developed a galectin-3 binding assay, and we found that it bound to integrins alpha V, beta 1, beta 5, and beta 6 in a concentration-dependent and glycosylation-dependent manner. So if we deglycosylated the integrins, which it has a number of, all the integrins have a number of different glycosylation sites. If we deglycosylated them, we abolished the binding of galectins to all 3 integrins. And if we only partially deglycosylated the O-linked glycosylation sites, this was partially inhibited. And so if we inhibited the galectin carbohydrate binding domain, specifically, we were able to reduce alpha V, beta 1, beta 5, and beta 6 binding to galectin-3, suggesting to us that the galectins prevented or galectins bound to the integrins. They also bind to TGF-beta and effectively stick the molecules together. Now, if you remember earlier, what I said was that for integrins to activate TGF-beta, there has to be close temporal and spatial interaction between these molecules. So it has to be activated, and then the integrin has to be sufficiently close to the TGF-beta receptor for the active TGF-beta released from the latent complex to interact with it. And you can see that under sort of some circumstances, if they're not close, this active molecule sort of drops in the gap between 2 stools. And so, and we also use plasma resonance imaging to confirm this, that if the galectins actually cluster the integrin and the TGF-beta receptor together, so that when it is activated, the active TGF-beta molecule can then drop into the arms of the receptor and then activate SMAD2 and SMAD3. So for this to occur, the basement membrane has to be disrupted, because the basement membrane is what normally keeps the epithelium away from the fibroblasts in normal lung, or certainly in normal alveolus. And so epithelial injury is crucial. And so these are data that just been accepted in Lancet Respiratory Medicine, looking at biomarkers of matrix and epithelial function in patients with pulmonary fibrosis, again from the PROFILE study. And we did a cluster analysis and defined 3 clusters. One, which reflects basement membrane collagen, so ProC28 and ProC4. A fascinating cluster of cross-linked fibrinogen, and then a cluster of 5 biomarkers which represent epithelial injury, so CIFRA 21-1, CA19-9, CA125, SPD, and MMP7. And these clusters were highlighted by a machine learning algorithm. These weren't pre-specified. And you'll also note that in the epithelial injury, so where you have high levels of epithelial injury, in this cluster, you also have lower expression, particularly of ProC4. So epithelial injury with low levels of ProC4 is bad. And certainly when you look at the Kaplan-Meier of these patients, they do much worse than patients in the other 2 clusters. So epithelial injury in the presence of failing basement membrane repair is a real problem. And you can see this temporally looking at these immunohistochemistry assessments of a patient without IPF and then 3 patients with IPF. And you can see that in the earlier stages of disease, the basement membrane is thickened. This is a collagen 4 immunohistochemistry stain. And you can see that there's thickening of the basement membrane, thickening of collagen 4, which tends to increase as the disease evolves, but at end stage fibrotic lung, all your collagen 4 disappears. And there is some physiological consequence to this. So we see that the earlier disease, you have you are still able to transfer gas across the basement membrane as measured by TLCO. So is there any more evidence to support basement membrane disruption? So we did some whole exome sequencing looking at rare coding variants, which threw up quite a few different variants using the PROFILE cohort, which is our longitudinal cohort of patients with IPF, and also the 100,000 genome cohort, which includes 111 patients with familial pulmonary fibrosis, as well as about 300 patients with sporadic pulmonary fibrosis. And as you can see, there's a large number of variants which were significant at the genome-wide significance threshold of 10 to minus 10. But I'm going to talk mainly about hemisentin-1, because that's a fascinating basement membrane protein. But if we looked at all of the genes together and did pathway analysis mapping, we found that the key genes which were identified were involved in actin cytoskeleton and G protein signaling, which are the pathways that I've been describing. So the actin cytoskeleton and cytoskeletal force is crucial, and as is G protein coupled receptor signaling through row A. So what about hemisentin-1? So it's a large extracellular protein. It's involved in basement membrane structure, and it binds to collagen 4. Furthermore, it's upregulated by TGF-beta, and variants in hemisentin-1 rare variants have been associated with age-related macular degeneration, which is fibrosis of the retina. So when we did functional annotation of the hemisentin-1 variants that we found in our burden testing analysis of whole exome sequencing, we found a large number of variants across different regions, and these are the different modular structures of hemisentin-1. And many of these are missense variants, so they don't all necessarily lead to obvious protein structural change. But one of them, the C4625W variant, led to a change in TGF binding in the thrombospondin-1 domain, which was very exciting. So you see here in the wild type that this is the latency-associated peptide, and the latency-associated peptide binds to the TSP region, the thrombospondin-1 domain of hemisentin, and that could affect TGF-beta activation. But in the mutant that's been associated with pulmonary fibrosis, the hemicentrin 1 binds the mature TGF-beta, so it doesn't bind the latent peptide, it binds the mature protein itself, which might lead to perpetual activation. This is a hypothesis which is yet to be tested. And the other thing we know is that matrix stiffness is crucial. And so we've been doing some experiments with gelatin and methacrylate gels, so called a gelma, and these can be freeze-dried and photosensitized to create a solid form, which is handy because you can then post this around the world if you need to. And you can tune the stiffness of the matrix, and more importantly, you can conjugate the matrix with matrix-associated peptides, and we've been focusing on basement membrane ones, so laminin and collagen 4. And what we see is that if we culture epithelial cells on the gelma, that under normal circumstances, you get increased cell death as the matrix stiffens. So if you think about fibrosis, and this is one of the key pathological observations in pulmonary fibrosis, that you get epithelial apoptosis in IPF. So if we increase the stiffness of the matrix to reflect the matrix that we see in pulmonary fibrosis and just culture epithelial cells on them, epithelial cells die or apoptose, which is consistent with what happens in disease. But if you conjugate laminin, and the blue is the scrambled peptide control, you can protect against this epithelial cell death. Again, illustrating the importance of the basement membrane in the development of pulmonary fibrosis. So this is putting all this information together. This is our current working hypothesis of how injury goes wrong to lead to the development of pulmonary fibrosis. So under normal circumstances, you have the alveolar epithelium sitting on an intact basement membrane, and on the other side of that basement membrane, you have fibroblasts secreting enough matrix just to keep everything stuck together. If there is mild injury or normal injury repair process, you get signaling through G protein coupled receptors through the pathway I described. You activate a little bit of TGF-beta, which is regulated by ALK1. There's some upregulation of integrins, but it can't activate any further TGF-beta because the basement membrane is in the way. However, in severe injury, you lose with loss of ALK1 or amplification of ACAP13, or indeed both. In the presence of a disrupted, unintact basement membrane, you get amplification of the Rho-ROCK pathway following injury because you have more ACAP13. This activates more TGF-beta. This is not de-repressed, which leads to increased and high levels of alpha-v beta-6 integrin on the epithelial surface, which can then directly interact with TGF-beta, which is either sequestered in the matrix or on the fibroblast surface. This then signals through the fibroblast and acts on the alpha-v beta-1 integrin, which in the presence of high levels of galectin-3 clusters them together and activates more TGF-beta in the fibroblast. And so you get this sort of positive feedback loop of enhanced TGF-beta activation without any of its normal regulatory processes. And this leads to the sort of snowball-like progressive fibrosis that we see in patients with advanced fibrotic lung disease. So can we find evidence of this in situ in patients with pulmonary fibrosis? So what we've started doing is spatial transcriptomics to try and identify what populations of cells there are in patients with IPF and how these interact. So we've used cores of lung tissue from patients with IPF and compared them initially with control So you can see those here. And then the spatial transcriptomics analysis revealed 4 distinct cells that are only observed, have a transcriptional profile that are only really observed in fibrotic tissue. And these include fibroblasts, fibrotic B lymphocytes, fibrotic macrophages, and fibrotic epithelial cells. And you can see where these cells are located. So the green cells here are the fibrotic epithelial cells. The brown cells are the fibrotic B cells, which don't seem to interact particularly with any other cell types. Macrophages, interestingly, which are often in the luminal surface of the honeycomb cyst. So these dark blue cells are the fibrotic macrophages. And then the fibrotic fibroblasts are the purple cells here, and you can see that they're interacting quite closely with the epithelium because the basement membrane is no longer intact. And there are very few of these cells in normal lung. And when we look at the cell interaction analysis, generally what we can see is that all cells in fibrotic tissue are highly transcriptionally active, even the cells that you would normally find in normal lung. They're there in fibrotic lung, but they're more transcriptionally active, and they're interacting with more of these of their neighboring cells or cells they wouldn't normally interact with. So in normal lung, you see there's the size of the dot and the number of the sort of colors in the fans reflects how many other cells they're interacting with. So you can see in normal lung that you have macrophages and dendritic cells are interacting with other cells, as are AT2 cells and club cells, but the vast majority of cells within the lung aren't doing a lot of interaction. Whereas in the fibrotic lung, pretty much all the cells are interacting with each other. And there are these unique cells, as I described, so the epithelial cell, fibroblast, macrophage, and B cell. And when we look at how they interact, so this is another core. And again, this is immunohistochemistry labeling of the cells. You can see that the fibrotic epithelial cells line the luminal surface, but not always. You have some here, there's no luminal surface for these fibrotic epithelial cells. They interact most commonly with fibrotic fibroblasts directly, and then the B cells are sort of coordinating things in a paracrine fashion. And using cluster analysis, that we've identified that there are 3 distinct fibrotic niches. One which involves primarily B cells, macrophages, and so fibrotic macrophages and alveolar macrophages. A fibrotic macrophage niche on its own, this is the SP1 niche, and then a matrix-derived fibrotic niche. And these are the transcriptional profiles that they have. Interestingly, by spatial transcriptomics, the fibrotic epithelial cells tend to cluster with bronchial epithelial cells, and which may give us some hint as to the origin of these cells, which is still quite controversial. And the fascinating thing is what are these fibrotic B cells doing? Because these are a cell that haven't really had much attention in the pathogenesis of pulmonary fibrosis, although there is emerging literature suggesting that these may be important. And the key difference about fibrotic B cells with other types of B cells is that you'll see that this is them here. So from a transcriptional perspective, they share features with plasma B cells, but importantly, they secrete or they synthesize high levels of decorin, and decorin is a very important matrix molecule, which is associated with regeneration, repair, promoting autoimmunity, and interacts closely with TGF-beta. So we're fascinated to progress the work trying to understand what the role of decorin synthesizing B cells is in fibrosis. So I'm going to finish there and just hopefully I've convinced you that pulmonary fibrosis is a progressive and fatal disease due to alveolar injury in a genetically susceptible person. That alveolar injury leads to G protein coupled signaling, which activates TGF-beta via the alpha-V beta-6 integrin and also the alpha-V beta-1 integrin. That dysregulation of integrin-mediated TGF-beta activation can occur at many levels, and they all promote fibrogenesis. And crucially, a normal functioning basement membrane is vital for alveolar repair. And I think this is one of the reasons why we can see complete destruction of the lung in things like COVID and pneumonia, and the patients can repair virtually normally, but in IPF, this doesn't happen. And that the fibrotic niche is highly transcriptionally active and highlights novel signaling pathways in unique fibrotic cells. So I'd like to thank my group who've done all this work, my collaborators, particularly Louise Wayne from Leicester, genetic epidemiologist, and I'd be happy to answer any questions. Thank you.
Thank you very much, Gisli. That was a fantastic lecture. I really, really enjoyed it. There aren't any questions in the Q&A at the moment, but I encourage everybody who's listening to please type in their questions. And in the meantime, I'll start off with ones. I think, yeah, as I said, fantastic lecture, huge amount of data there. And I'm just wondering, is this process of interstitial fibrosis one that is almost like a default mechanism which could occur in whatever organ, whether it's, for example, the liver or, okay, you've spoken about the lung, but in other organs, is this, do you think, a sort of a similar, a common process that could be targeted in other diseases?
Absolutely, I think that's a great question, and certainly it's that was the sort of fundamental principle underlying our strategic priority grant awarded by the MRC and the NIHR to look at fibrosis in multiple organs. And we pitched it on that principle that these are common mechanisms across organs, and you will see different ways of TGF-beta activation in all organs. And we have evidence from animal models, for example, that liver fibrosis is very much driven by alpha-V beta-1. And some of our knockout studies have shown that, for example, ELK1 knockout mice get liver fibrosis and steatohepatosis. And certainly when you look at the comorbidities, there are clusters of comorbidities that do come up with IPF. So classically, systemic hypertension, half of patients with IPF have systemic hypertension. And there are other sort of different clusters, chronic kidney disease, for example. So indeed, when we've looked at a range of organ cohorts, many patients, in fact, only a third of patients have single organ chronic kidney fibrosis, chronic liver fibrosis, or chronic lung fibrosis. And the vast majority will have at least 2 organs, and many 3 or even 4. And we've looked in UK Biobank as well. And one of the issues we found is that different specialists have different ways of describing fibrosis. So, for example, the neurologists largely don't believe fibrosis exists. They just call it something different, but the mechanisms are very similar. So nomenclature has been a challenge. And we were thinking a bit like cancer. You know, there are hallmarks of cancer. Even though cancer is not always the same in every organ, there are hallmarks that make it distinct. But fibrosis we haven't come together as a community to actually organize our thoughts and our nomenclature to make it more obvious. And I think that's really important, because when we looked at codes across death certificates, it's 40% of people have a fibrotic disease code on their death certificate, which is quite astonishing.
Yeah. No, I think you're right. We probably should join forces. It seems to be a similar pathogenic process where we should probably be more consistent with the nomenclature and also share our findings. Obviously there are very desmoplastic cancers, and a lot of people are working on that. But in the meantime, sorry, there are 4 questions that have come in, so I shouldn't hog you in terms of asking you questions. So the first one is, does the respiratory microbiome have a role?
That's a great question, and the answer is yes. But how precisely it has a role, it's hard to say. It's clearly, and there is an interaction between the microbiome and the alveolar epithelium. And there's some interesting data published out of Imperial, actually, and North America, looking at the effects of the microbiome and the development of fibrosis. But that's very much the luminal side. So is it acting as an injury? It does altering the microbiome, alter the injury repair response. So is it one of the many injuries that occurs that in some people is going to not be a problem, but in others will be a problem? Because when people have looked at lung tissue itself, so Danny Cass and colleagues from Pittsburgh did this, they actually took the lung rather than lavage and found very little evidence of microbiome involvement. So I think it might be more acting as the microbiome changes the injury rather than the response.
Right. Okay. So a question related to COVID. COVID virus-induced fibrosis, could this resolve later by partial fibrosis and resolving of symptoms?
So COVID is fascinating. We're sort of finalizing some of our molecular and cell biology work because the SARS-CoV-2 has an RGD domain, which means it can bind, and it does in vitro bind integrins. But also ACE2 has an RGD binding domain, and both of them have cryptic RGD domains, which means that something has to happen to reveal the RGD domain. And what it appears to be is that once the SARS-CoV-2 virus binds ACE2, it actually the cryptic RGD domain on the ACE2 molecule is then able to interact with the integrin and increases internalization. And that's why patients with pulmonary fibrosis did so badly in the first wave of COVID, because they had high levels of beta-6 integrin, and they got a lot more alveolar injury, and the mortality rates in patients with pulmonary fibrosis in the first couple of waves was eye-watering. What that means long-term, we don't know yet. What we do know is that from our analysis is that patients who have persisting evidence of fibrosis have high levels of MMP7, and those data are currently under review, and the preprint is available. And things don't seem to get better after 6 months. So what you're left with at 6 months is what you're left with forever. And it may be that if you have the right genetic predisposition to longer term fibrosis, that things may actually start to get worse, and you get the sort of autonomous fibrosis that we see in progressive disease. So I think COVID is fascinating because it's a natural experiment. You've given the whole world an injury at the same time. And we're going to see what the long-term consequences of that are, but it is going to take us 20 years to figure it out.
Right. Okay. Thank you. So a question from Charles Parker. He's returning now a bit to the next 2 questions to do with genetics and also the transcriptomics. So as well as the germline SNPs you spoke about, have you found any evidence of acquired somatic mutations?
No, but we haven't looked especially hard. So the one thing we have looked at for somatic mutations is mosaic loss of Y, which is so that's the commonest sort of aneuploidy event, which would be a pre-full somatic event. And there is evidence of mosaic loss of Y in IPF, and again, this manuscript just being prepared for publication. But interestingly, mosaic loss of Y is also an age-dependent phenomenon, and we didn't find any direct causal relationship. So what it appears to be is that the telomere mutations, which are major drivers of IPF, seem to be causing both IPF and mosaic loss of Y. So we see mosaic loss of Y associated with IPF, but it's not directly causal.
Right.
And we haven't looked beyond that.
Okay, thank you. One from Marnix Janssen. Can he ask, with regards to the spatial transcriptomics and the dynamics of the disease, how do you make sure the cause represent early stage? And more generally, how difficult is it to get early stage material from these patients?
So that's, again, a wonderful question, and a plea, if I may. It's really difficult. It's really difficult to get any tissue from patients with pulmonary fibrosis. I think it's possibly, of all the things I do in my life, the hardest thing to do. Yeah, and I've cycled 200 miles. I've cycled up hills, mountains, and none of this is as hard as trying to get tissue from patients with pulmonary fibrosis, let alone early stage. So what we're actually thinking of doing now is actually taking control tissue. So actually not even worrying whether they have fibrosis or not, but we can genotype it. So we'll have an at-risk population versus a non-at-risk population. And then we can injure it in vitro and see how they respond, which will probably be the best representation of really early disease, because even with the rare variants, they don't manifest disease often until early 40s. But yeah, trying to get different stages of fibrosis is virtually impossible, because we'll either get weird disease from a diagnostic biopsy, and people only have diagnostic biopsies if there's something quirky about their presentation, or post-mortem or explant, transplant, which is all late stage. So yeah, it's a weakness of these studies.
Yeah. Okay, we've got two last questions. See if we can get through them in the last 2 minutes. What is the trigger mechanism for the fibrogenic response associated with pneumonitis?
So I think it'll probably be a combination of, definitely the genetic background. So they don't repair. And we see this with pollution. The pollution data is quite terrifying, actually. If you look at PM2.5 in patients without genetic risk factors, the risk of fibrosis is about 10%, which is, not going to set any sort of politicians on fire. But if you look at patients with MUC5B, the major, just MUC5B, the risk of fibrosis in these people is over 300% greater. So genetic response is crucial. The trigger is the alveolar injury. So, and we can see this with that biomarker data. In COVID, we see this too, that the fibrosis is associated with high levels of MMP7 and SPD. So we think you get alveolar injury either recurrently or persistently in a genetically predisposed population. But we think it's inhaled. So I guess the trigger is whatever you breathe in promotes epithelial injury. Actually, the other one which we've shown to be important is aspiration of gastric contents. So anything that gets into your lungs which can injure the epithelium is a trigger.
And I guess that leads on to the very last question: is can pet hair induce fibrosis?
Can pet hair? We've never seen an association with cats and dogs. The pets that do it are birds, parrots and pigeons, and that's more immunogenic. So it's an immunogenic. So I suspect pet hair is just too big. So it's not asbestos fibers are small enough to get down there, I don't think.
Yeah.
So I suspect not.
Right. Well, thank you very much. Mark has a tradition of, finishing up these Grand Round lectures on time at 10 o'clock, and the fire alarm just went off here, indicating to me it's 10 o'clock. That's a practice fire alarm, by the way. So thank you very much for your fantastic lecture. I certainly learned a lot, and I'm sure all of the attendees did as well. So thank you, and we'll be in touch.
Thank you very much for the invite. It's a pleasure to speak to you all. Thank you.
Bye then. Bye.